Class 11, Chapter 9, Biomolecules. There is a wide diversity in living organisms in our biosphere. Now, a question that arises in our mind is, are all living organisms made up of same chemicals, that is, elements and compounds? You have learned in chemistry how elemental analysis is performed. If we perform such an analysis on plant tissue, animal tissue, or microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several others, and their respective content per unit mass of living tissue. If the same analysis is performed on a piece of earth crust as an example of non-living matter, we obtain a similar list. What are the differences between the two lists? In absolute terms, no such difference could be made out. All the elements present in the sample of earth crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than in the earth's crust. Now, how to analyze chemical composition? We can continue asking in the same way, what type of organic compound are found in living organism? How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or a piece of liver, etc., and grind in trichloroacetic acid. Using a mortar and a pestle, we obtain a thick slurry. If we were to strain this through a cheesecloth or cotton, we would obtain two fractions. One is called the filtrate, or more technically, the acid soluble pool, and the second retentate, or the acid insoluble fraction. Scientists have found thousands of organic compounds in the acid soluble pool. In higher classes, you will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that one extracts the compound, then subjects the extract to various separation techniques till one has separated a compound from all other compounds. In other words, one isolates and purifies a compound. Analytical techniques, when we apply to the compound, give us an idea of the molecular formula and the probable structures of the compound. All the carbon compounds that we get from the living tissue can be called biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One way, a small amount of living tissue and dry it. All the water evaporates. The remaining material gives dry weight. Now, if the tissue is fully burned, all the carbon compounds are oxidized to gaseous form and are removed. What is remaining is called ash. This ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid soluble fraction. Therefore, elemental analysis gives elemental composition of animal tissue in the form of hydrogen, oxygen, chlorine, carbon, and etc. While analysis for compounds gives an idea of this kind of organic and inorganic constituents present in the living tissue. From a chemistry point of view, one can identify fractional group like aldehyde, ketone, aromatic compound, etc. But from a biological point of view, we shall classify them into amino acid, nucleotide base, fatty acid, etc. Amino acid are organic compounds containing an amino group and an acidic group as substituent on the same carbon, that is the alpha carbon. Hence they are called alpha amino acid. They are substituted methanes. There are four substituent groups occupying the four valency positions. These are hydrogen carboxyl group, amino group, and a variable group designated as R group. Based on the nature of R group, there are many amino acids, alkyl group. However, those which occur in proteins are only of 20 types. The alkyl group in these proteinaceous amino acids could be a hydrogen. The amino acid is called glycine. A methyl group, alanine, hydroxyl methyl serine, etc. 3 of the 20 are shown in the figure below. The chemical and physical properties of amino acids are essentially of the amino, carboxyl and alkyl functional group. Based on the number of amino and carboxyl groups, there are acidic glutamic acid, basic lysine and neutral valine amino acids. Similarly, there are aromatic amino acid, tyrosine, phenylalanine, tryptophan. A particular property of amino acid is the ionizable nature of NH2COOH group. Hence, in solution of different pH, the structure of amino acid changes. Lipids are generally water insoluble. They could be simple fatty acids. A fatty acid has a carboxyl group attached to an alkyl group. The alkyl group could be a methyl or ethyl or higher number of CH2 group. 1 carbon to 19 carbon. For example, palmitic acid has 16 carbon including carboxyl carbon. Arachidonic acid has 20 carbon atoms including the carboxyl carbon. Fatty acid could be saturated without double bond or unsaturated with one or more double bond. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acid. Here the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglycerides and triglycerides. These are also called fats and oils based on the melting points. Oil have lower melting point, example gingerly oil, and hence remain as oil in winters. Can you identify a fat from market? Some lipids have phosphorus and phosphorylated organic compounds in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissue, especially the neural tissue, have lipids with more complex structures. Living organisms have a number of carbon compounds in which heterocyclic ring can be found. Some of these are nitrogen base, adenine, guanine, cytosine, uracil and thymine. When found attached to a sugar, they are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called nucleotides. Adenosine, guanosine, thymidine, uridine and cytidine are nucleosides. Adenalic acid, thymidalic acid, guanalic acid, uridalic acid and cytidalic acids are nucleotides. Nucleic acids like DNA and RNA consist of nucleotides only. DNA and RNA function as a genetic material. So these are the figures where we found some structures: cholesterol, phospholipid, adenine, adenosine, uridine, etc., etc. Now, primary and secondary metabolites. The most exciting aspect of chemistry deals with the isolating thousands of the compounds, small and big, from living organisms, determining their structures and if possible synthesizing them. If one were to make a list of biomolecules, such a list would have thousands of organic compounds including amino acids, sugars, etc. For reasons that are given in section 9.10, we can call them the biomolecules as a metabolites. In animal tissue, one notices that the presence of all such categories of compounds shown in figure there are called primary metabolites. However, when one analyzes plant, fungal and microbial cells, one would see thousands of compounds other than these called primary metabolites. Example: alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigment, scents, gums, spices. These are called secondary metabolites. While primary metabolites have identifiable function and play known role in normal physiological process, we do not at the moment understand the role or function of all the secondary metabolites in host organism. However, many of them are useful to human welfare. Example: rubber, drugs, spices, scents and pigments. Some secondary metabolites have ecological importance. In the later chapter and years you will learn more about this. Biomacromolecules. There is one feature common to all those compounds found in the acid soluble pool. They have molecular weight ranging from 18 to around 800 Dalton approximately. The acid-insoluble fraction has only 4 types of organic compounds, that is protein, nucleic acid, polysaccharides and lipids. These classes of compounds, with the exception of lipids, have molecular weight in the range of 10,000 Dalton and above. For this very reason, biomolecules, that is chemical compounds found in living organisms, are of two types. One, those which have molecular weight less than 1000 Dalton and are usually referred to as macromolecules or simply biomolecules. While those which are found in the acid insoluble fraction are called macromolecules or biomacromolecules. The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Then why do lipids, whose molecular weight do not exceed 800 Dalton, come under the acid insoluble fraction, that is macromolecular fraction? Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structures like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicles get separated along with the acid insoluble pool, and hence in the macromolecular fraction, lipids are not strictly macromolecules. The acid-soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelle become the acid-insoluble fraction. Together they represent the entire chemical composition of living tissue or organisms. In summary, if we represent the chemical composition of living tissue from the abundance point of view and arrange them class-wise, we observe that water is the most abundant chemical in living organism, 17 to 90% of total cellular mass. Protein. Protein are polypeptide. They are linear chain of amino acid linked by the peptide bond as shown in the figure. Each protein is a polymer of amino acids. As there are 20 types of amino acid: alanine, cysteine, proline, tryptophan, lysine, etc. Cysteine. Okay. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n number of times. This information about the amino acid content is most important as later in your nutrition lesson. You will learn that certain amino acids are essential for our health and they have to supply it through our diet. Hence, the dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make. While we get essential amino acids through our diet or food, protein carry out many functions in living organisms. Some transport nutrients across cell membranes, some fight infectious organisms, some are hormones and some are enzymes, etc. Collagen is the most abundant protein in the animal world, and ribulose bisphosphate carboxylase oxygenase Rubisco is the most abundant protein in the whole biosphere. Collagen most abundant in animal world and Rubisco most abundant in whole biosphere. Polysaccharide. The acid insoluble pellet also has polysaccharide carbohydrates as another class of macromolecules. Polysaccharides are long chain of sugar. They are threads, literally a cotton threads, containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharides, that is glucose. Cellulose is a homopolymer. Starch is a variant of this but present as an storehouse of the energy in plant tissue. Animals have another variant called glycogen. Inulin is a polymer of fructose in a polysaccharide chain, say glycogen. The rigid right end is called the reducing end and the left end is called non-reducing end. It has branches as shown in the form of cartoons. Starch form helical secondary structures. In fact, starch can hold I2 molecules in the iodine molecules in the helical portion. The starch I2 is blue in color. starch iodine, sorry. Cellulose does not contain complex helices, hence cannot hold iodine molecule. Plant cell walls are made up of cellulose, paper made up of plant pulp, and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have as building blocks amino sugars and chemically modified sugars. Example glucosamine and acetylgalactosamine, etc. Exoskeleton of arthropods, for example, have a complex polysaccharide called chitin. These complex polysaccharides are mostly homopolymers. Nucleic acid. The other type of macromolecule that one would find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides together with polysaccharides and polypeptides. These comprise the true macromolecular fraction of any living tissue or cell. For nucleic acid, the building block is a nucleotide. A nucleotide has a three chemically distinct component. One is a heterocyclic compound, and the second is a monosaccharide, and the third a phosphoric acid or phosphate. As you notice in figure, the heterocyclic compound in nucleic acid are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are substituted purines while the rest are substituted pyrimidines. The skeletal heterocyclic ring is called as purine and pyrimidine respectively. The sugar found in the polynucleotides is either ribose, monosaccharide pentose, or 2-deoxyribose. A nucleic acid containing carbodeoxyribose is called deoxyribonucleic acid, that is DNA, while that which contain ribose is called ribonucleic acid, that is RNA. Structures of protein. Protein, as mentioned earlier, the heteropolymer containing strings of amino acid. Structures of molecules mean different things in different contexts. In inorganic chemistry, the structure invariably refers to the molecular formula, example sodium chloride, magnesium chloride, etc. Organic chemists always write a two-dimensional view of the molecules while representing the structures of the molecules, example benzene, naphthalene, etc. Physicists conjure up the three-dimensional views of a molecular structures, while biologists describe the protein structure at four levels. The sequence of amino acid, that is positional information in a protein, which is the first amino acid, which is second, and so on, is called the primary structures of the protein. A protein is imagined as a line, the left end represented by the first amino acid and the right end represented by the last amino acid. The first amino acid is also called as N-terminal amino acid. The last amino acid is called C-terminal amino acid. A protein thread does not exist throughout as an extended rigid rod. The thread is folded in the form of a helix similar to a revolving staircase. Of course, only some portion of the protein thread are arranged in the form of a helix. In protein, only right-handed helices are observed. Other regions of the protein thread are folded into other forms in what is called the secondary structures. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structures. This gives us a three-dimensional view of protein. Tertiary structures is absolutely necessary for the many biological activities of protein. Some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individuals folded polypeptides or subunits are arranged with respect to each other, example linear strings of spheres arranged upon each other in the form of a cube or plate, etc., is the architecture of a protein, otherwise called the quaternary structures of a protein. Adult human hemoglobin consists of four subunits. Okay, two of these are identical to each other. Hence, the two subunits of alpha type and two subunits of beta type together constitute the human hemoglobin. Enzymes. Almost all enzymes are protein. There are some nucleic acids that behave like enzymes. These are called ribozymes. One can depict an enzyme by a line diagram. An enzyme, like any protein, has a primary structure, that is amino acid sequence of the protein. An enzyme, like any protein, has the secondary and the tertiary structures. When you look at the tertiary structures, you will notice that the backbone of the protein chain folds upon itself. The chain crisscross itself, and hence many crevices or pockets are made. One such pocket is active site. An active sites of an enzyme is a crevice or pocket into which the substrate fits. Thus, enzymes through which their active sites catalyzes reaction at high rate. Enzymes catalyst differs from inorganic catalyst in many ways, but one major difference need mention. Inorganic catalysts work efficiently at high temperatures and high pressures, while enzymes get damaged at high temperatures. However, enzymes isolated from organisms who normally live under extremely high temperatures, example hot vents and sulfur springs, are stable and retain their catalytic power even at high temperatures. Thermal stability is thus an important quality of such enzymes isolated from thermophilic organisms. Chemical reaction. How do we understand these enzymes? Let us first understand the chemical reaction. Chemical compounds undergo two types of changes. A physical changes simply refers to a change in shape without breaking of bonds. This is a physical process. Another physical process is change in state of matter. When ice melts into water or when water becomes a vapor, these are also physical process. However, when bonds are broken and new bonds are formed during transformation, this will be called a chemical reaction. For example, barium hydroxide H2O SO4 BaSO4 2H2O is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Rate of physical or chemical process refers to the amount of product formed per unit time. It can be expressed as rate product per unit time. Rate can be called the velocity if the direction is specified. Rate of physical and chemical process are influenced by temperature among other factors. A general rule of thumb is rate doubles or decreases by half for every 10 degrees Celsius change in either direction. Catalyzed reaction proceed at rates vastly higher than that of uncatalyzed one when enzymes catalyzed reaction are observed, and the rate would be vastly higher than the same but catalyzed reaction. In the absence of any enzyme, this reaction is very slow, with about 200 molecules of H2CO3 being formed in an hour. However, by using the enzymes present with the cytoplasm called carbonic anhydrase, the reaction speed dramatically with about 6 lakh molecules being formed every second. The enzymes has accelerated the reaction rate by about 10 million times. The power of enzyme is incredible indeed. There are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of these steps is catalyzed by the same enzyme complex or different enzymes, is called metabolic pathway. Is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzymes catalyzed metabolic reaction. When you study respiration in chapter 12, you will study the reaction. At this stage, you should know that this very metabolic pathway with one or two additional reaction gives rise to a variety of metabolic end products. In our skeletal muscles, under anaerobic condition, lactic acid is formed. Under normal aerobic condition, pyruvic acid is formed. In yeast, during fermentation, the same pathway leads to the production of ethanol, alcohol. Hence, in different conditions, different products are possible. So now, how do enzymes bring about such high rates of chemical conversion? To understand this, we should study enzymes a little more. We have already understood the idea of active site. Now the chemical or metabolic conversion refers to a reaction. The chemical which is converted into product is called a substrate. Hence enzymes, that is protein with three-dimensional structures including an active site, convert a substrate into product Symbolically, this can be depicted as: it is now understood that the substrate has to bind the enzyme at the active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of an S complex. E stands for enzyme and S for substrate. This complex formation is a transient phenomenon. During the state where substrate is found to the enzyme active site, a new substrate of the substrates called transition state structure is formed. Very soon after the expected bond breaking making is completed, the product is released from the active site. In other words, the structure of substrates gets transformed into the structure of products. The pathway of this transformation must go through the so-called transition state of structures. There could be many more altered structure states between the stable substrates and products. Implied in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to energy status of the molecule of or the structure. Hence, when we look at this pictorial through a graph, it looks like something as in figure given. The y-axis represents the potential energy content, the x-axis represents the progression of the structural transformation or states through the transition state. You would notice two things: the energy level difference between substrate and product, and if product is at lower level the substrate than substrates, the reaction is an exothermic reaction. One need not supply energy by heating in order to form the product. However, whether it is an exothermic or a spontaneous reaction or an endothermic or energy requiring reaction, the substrate has to go through as much higher energy state or transition state. The difference in average energy content of substrates from this transition states called activation energy. Enzymes eventually brings down this energy barrier, making the transition of substrate to product more easy. Nature of enzyme action. Each enzyme has a substrate binding sites in its molecule, so that a highly reactive enzyme substrate complex is produced. This complex is short-lived and dissociates into its products. And the unjoined enzyme with an intermediate formation of the enzyme product complex. The formation of enzyme complex is essential for catalysis. The catalytic cycle of enzymes action can be described in the following steps. First, the substrates bind to the active site of the enzyme, fitting into the active site. Second, the binding of the substrates induce the enzyme to alter its shape, fitting more tightly around the substrates. Third, the active site of the enzyme, now is close proximity of the substrates, breaks the chemical bonds of the substrates and the new enzyme product complex is formed. And the fourth, the enzyme release the products of the reaction and free enzyme is ready to bind to another molecule of the substrates and run through the catalytic cycle once again. Now, factors affecting enzyme activity. The activity of enzyme can be affected by change in the condition which can alter the tertiary structures of the protein. These include temperature, pH, change in substrate concentration, or binding of specific chemicals that regulate its activity. Temperatures and pH. Enzymes generally functional in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature and pH, called the optimum temperature and optimum pH. Activity declines both below the and above the optimum level. Low temperatures preserves the enzyme in the temporarily active state, whereas the high temperature destroy enzymatic activity because proteins are denaturated by heat. Concentration of substrate. With the increase in substrate concentration, the velocity of enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate's molecules, and after saturation of these molecules, there are no free enzymes molecules to bind with the additional substrate molecules. The activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of the chemical shuts off enzyme activity, the process is called inhibition and the chemical is called inhibitor. when the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of enzyme. It is known as competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrates for the substrate's binding sites of the enzyme. Consequently, the substrates cannot bind, and as a result, the enzyme action declines. Example: inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogens. Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the types of reaction they catalyze. Enzymes are divided into 6 classes, each with 4 to 13 subclasses and named accordingly to by 4-digit number. Oxidoreductase dehydrogenase. Enzymes which catalyze oxidoreduction between two substrates. Example: as reduced, as oxidize, and reduce. Transferase. Enzyme catalyzing a transfer of a group other than hydrogen between a lone pair of substrate and substrate dash. Hydrolases. Enzyme catalyzing hydrolysis of ester, ether, peptide, glycosidic, carbon-carbon, carbon halide, or phosphorus and nitrogen bonds. Lyase. Enzyme that catalyze removal of groups from substrates by mechanism other than hydrolysis, leaving double bonds. Isomerase, including all enzymes catalyzing interconversion of optical, geometrical, or positional isomers. Ligases. Enzymes catalyzing the linking together of two compounds. Example: enzymes which catalyze joining of carbon oxygen, carbon sulfur, and carbon nitrogen, phosphorus oxygen, etc. Bonds. Cofactor. Enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which no protein constituents called cofactor are bound to the enzymes to make the enzymes catalytically active. In this instance, the protein portion of the enzymes are called the apoenzyme. Three kinds of cofactors may be identified: prosthetic groups, coenzymes, and metal ions. Prosthetic groups are organic compounds and are distinguished from the other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen. Heme is the prosthetic group and it is a part of the active site of the enzyme. Coenzymes are also organic compounds, but their association with the apoenzyme is only transient, usually occurring during the course of the catalysis. Furthermore, coenzymes serve as cofactor in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins. Examples: coenzymes nicotinamide adenine dinucleotide (NAD) and NADP contain the vitamin niacin. A number of enzymes require metal ions for their activity, which form coordination bonds with side chain at the active site and at the same time form one or more coordination bonds with the substrates. Example: zinc is a cofactor for the proteolytic enzymes carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzymes. So that's it. Thank you so much.